This is a production of Cornell University.
Perfect. So today it's going to be sort of like an overview of the new technologies and new things that have learned about apple rootstocks since they were initially utilized in horticulture or pomology. So these are new technologies of the apple rootstock breeding program. So this is where we come from. We have the Plant Genetic Resources Unit, and the apple rootstock breeding program now is sort of like a collaboration between several different research programs at Agritech. And right here, let me see if I can get, there we go, a pointer. Let's do laser pointer. Right here, you have Jim Cummings and Herb Allwinkle. And I sit on the shoulders of very great people because they are the inceptors of the breeding program more than 50 years ago. In fact, I was not even a twinkling in the eye of my parents when this started. So, and then this is Terrence Robinson, who joined the breeding program later on, but for more than 40 years. And he is not just a preacher, but he's actually not a talker, but actually he walks the walk. So he has his own orchard, and these are a bin of Honeycrisp apples, 100%, 99% great, no bitter pit, and they're grown in G969 rootstock. And this is Abais Khan. He helps us with the fire blight research and in general disease resistance research. I'd like to recognize Charlie. He passed away a few years ago, but the trees that he planted are still in the ground. This is Todd Holleran right here, who is a technician with us, and Sarah Bauer, who is also a technician with us. But we've had many, many people. Okay, I gotta do this. We've had many, many people work with us in the breeding program. So I just like to show pictures of all the people that in one way or another contributed to our breeding program. So Bill mentioned this. This is a partnership. Our breeding program is a unique partnership between the USDA and Cornell University, and it has resulted in the Geneva rootstocks, which are grown worldwide. We're in South Korea, Brazil, Kazakhstan, you name it, wherever they grow apples, and we're there. So I mentioned a little bit about the historical perspective. So this program started in 1968. Dr. Cummings retired right around this time, and then right in late 1990s, early 2000s, the USDA-ARS took the lead of the breeding program, providing a scientist, and we've released several rootstocks in that process. And the production right now of rootstocks is about 15 million. It goes up and down. 15 million trees planted every year, maybe sometimes up to 30 million, depends on the glut or planting projects that are around the world. So we have more than 100 million trees in the ground, Emily, right? So Emily's right here from CTL, and I'll have a slide about them. So rootstocks, I hope all of you know what rootstocks are. We're just going to go very quickly. They are vegetatively propagated by stool bed or by some other methods, and this is what they look like when we harvest them. They got roots, and liner like that. They said they get sold in bundles like that. And then you take your favorite apple and you graft it onto the rootstock and you make trees. And so the rootstocks have now become a central actor in all of these interactions in the orchard. So we have interactions with the diseases, climate, soil, and then sometimes the orchard management or the type of things. So with rootstocks, you can do a lot. So years ago, we received a a national award, the rootstock breeding program and the technology transfer people competing against NASA and all skunk works and all sorts of other things. And that award was because of the number of field trials and the number of connections that we had with growers in order to transfer all the technologies that we develop into and put them into the hands of apple growers in the US. And so we've released several rootstocks. These are just a number of rootstocks that we released. Some of them have received medals because of the unique qualities that they have portrayed. And in fact, this was the first rootstock that combined both replant resistance, resistance to woolly apple aphid, and all sorts of other good things. But no rootstock is perfect, so we continue to work on things. So this is a table that sort of describes most of the rootstocks that we have released so far, and it gives you an idea of all the different traits that we like to talk about that are unique with the rootstocks. Back when I started in 2001, this is what the market looked like for apple rootstocks. It was closely 90%, close to 90%, it was a monoculture of Malling-9 rootstock. And I'll talk a little bit about that. And now, this is for 2022, the landscape is very, very different. And we've increased the diversity of the rootstocks. And also, I think we also increased the resiliency of the whole industry. And diverse rootstocks have allowed us to do different things with training systems and with production and models and so on and so forth. So here, right here, you have Nicola Della Betta, who passed away a couple of years ago, but his nice planting of Golden Delicious apples in Trentino Alto Adige. This is in northern Italy, part of northern Italy. And look at how many branches are coming off. So this is a new, different type of system. And this is a system right here that is in Washington State. So in order to do that, you need different rootstocks, different abilities to provide architectures. And the other thing that we're doing, that we have been doing in the past few years, is because we have these many rootstocks, we're trying to match the rootstocks to the needs of the qualities of the science. So for example, we just have 3 that are well known: Fuji, Gala, Honeycrisp, and then we try to match the different characteristics. Like Fuji has too much vigor, so we need something that controls that vigor and things like that. Or Honeycrisp, there are fruit disorders, and so good calcium to potassium ratio in the fruit. And we're also coming up with, we have come up with this concept of the designer traits to manage these varieties, the new varieties, in a way that they become more productive and more remunerative. So we have increase in productivity, the branch angle modification, replant disease tolerance, and so on and so forth, and then unique nutrient uptake. In the hands of the growers, these different things can mean a lot of money. For example, the resistance to fire blight in New York growers and growers in most of the United States is like a must-have. If you don't have it, I've seen, and you'll see, I'll show you pictures in a few minutes. If you don't have it, you lose your entire orchard. You lose your production for many years. So we have these technologies, and they mean apples. So these are some of the new, describe the example of rootstock induced traits. So here's a rootstock, and it's inducing the trait on the scion that's attached. And the first one that was described was the dwarfing and early bearing. But then, the new ones are, and I'll have slides about them, are like branch angle modifications, more flowering above bud break in low chill environments, hormone balances that change because of the different rootstock that the sign is attached on, the mineral nutrient concentration, and then whole tree tolerance to fire blight, and there are more traits than that. So let's go with the first one, which is the most ancient one, the one that was developed. So here you go. This is the way 100 years ago trees were planted and harvested and grown. You see some nice ladders over there. And then this is an example of two trees planted at the same time. They were at the same size when they were planted. But one is just growing huge and the other one is like this little tiny thing and it's full of apples. And that, the only difference between those trees, the sign is the same, is the rootstock, okay? So the rootstock is that powerful to, change the behavior of the sign. And given that, you're able to have these types of growth architectures or in the field, and it's really industrial. This has become an industrial thing. So where did dwarfing come from? So in 1912, this fellow got together and he was the first DUS tester. He took all the rootstocks, apple rootstocks that were available in Europe, brought them into his research station in England, tested them out, and with the methods that they had at that time, he described exactly this series of rootstocks so that there were no duplications and things. And so this became the Malling series of rootstocks, of which Malling 9 is the most important. But dwarfing, where did it come from? Well, it was available, it was there a long time before this Rotten Hatton described it. And so this is a book from 1866 that talks about the Dutch creeper or the Doucin stock. Okay, I can go further back about 100 years in French, and you can see it talks about Doucin stock and Doucin le feu de paradis, and you can go even further back. So the dwarfing character has been there a long time. A while back, we described the genetics of how this dwarfing trait works. It's mostly due to two main loci in the apple genome, and this is where they are, chromosome 5 and chromosome 11. But in the way that you combine the alleles, you're able to produce rootstocks that will induce different architectures, different sizes and productivities of the attached scion. It can get more complicated than just two loci, but these are the two main ones. And so what that has done, it's taken those big, huge trees that we saw, and it's reduced them to sometimes, well, a lot of growers are doing this in a planar architecture, which is able to capture the sun in a much more efficient way, and also expose the fruit to better environments for that. This transformation is sort of like the green revolution of they induced the green revolution, just like wheat, the dwarfing wheat, or in tomatoes, what is it, the self-pruning locus. But when we look at the impact, the yearly impact of just these two loci, and this is being very conservative, it's about $2 billion a year for the US industry, for the US apple industry, because of the transformation of productivity given by the rootstocks, okay? That's $2 billion of increased efficiency every single year. And this is a calculation that I did based on statistical data from the USDA-ARS. Of course, we had a little bit of increase due to better management practice and fertilizer and so on and so forth, but the dwarfing has been the largest driver of this revolution. And nowadays, we are planting orchards that are being mechanically harvested, and this is an interesting harvester. But without those planar canopies, you cannot have robots that go like beep, 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 and pull apples off the tree without any or very little supervision. Pretty amazing. So it's been more than 50 years since this breeding program started, and it's taken a long time to release several upper rootstocks, and many of the rootstocks were released that were initially crossed in 1970s, 80s. So it takes decade-level research in order to study apple rootstocks. And so these are some of the long-term papers that we have produced. Were the research done by Terence Robinson and our group, and has been 15 years, sometimes 20 years, where we monitor the orchard. So this is the key to these to rootstock research really is long-term performance, because once the grower puts in that rootstock or that tree in, guess what? It is in for at least 12 years to 25 years. That's pretty much the life of the rootstocks. And so these are different manuscripts that we produced about this long-term research. We've also found that some of the new traits are traits that had been observed before, but because they didn't have enough diversity of rootstocks, they couldn't quite get it. Is the effect of fruit the rootstock on fruit quality. And you can think about, sun, hormones, water, and so on and so forth, and nutrients. And so I'll talk a little bit about that. But this is an example of Gala. This is one scion variety. We had it grafted on 129 rootstocks in this research. So I just picked some pictures of samplings. You can see the effect of on firmness, maturity, sugar. Well, you can see the sugar, but let me tell you, it does. And color, and fruit size, and so on and so forth. And so you go like, how is that happening? Well, okay, so we found that rootstocks, this is work done by John Lorden with us, and we show that rootstocks actually influence the scion variety, the amount of phytohormones that we can find there. And this is just an example of ABA, but it's cytokinins, auxins, all sorts of other things. And the other thing that the rootstocks are doing is sometimes our Geneva rootstocks are actually changing the architecture of the apple tree. And so we have noticed that several of our rootstocks will open up the architecture of different grafted scions. And that means that by doing that, they provide more, I guess, better architecture for sun, harvesting the sun. Sometimes also they provide better architecture for nursery trees and how they develop, and growers not having to tie off each individual branch quickly. Something else that we noticed in the past, I think we started this research back in 2010, 2009, where we said, let's look at how these rootstocks are affecting our science in terms of nutrient levels. And one of the things that we found was that, and it's strongly genetically inherited, is that some of our rootstocks will consistently increase the concentration of boron in the scion. And this is, you can see right here, over multiple years. This is from data from 2014 up to 2017, but I can tell you that it continues this on and on. And so, and that's true for phosphorus, calcium. It's a little bit different. It's true so much that we did a larger genetic study, and we found several QTLs. This is the first of its kind, I think, in the tree world, in the pomology world, where we actually found root systems that were modulating, and we learned about the genetics of those. Very simple thing. How do these look like in the field? So this is the genetics of nitrogen, at least in the population that we were studying. And so two loci for that are modifying the nitrogen uptake, one on chromosome 2, one on chromosome 14. But this is what it shows up in the field, okay? And I'm gonna move a little bit right here. Can you squint your eyes and see what's happening right here in the middle? These are two different rootstocks, 213 on that side and 214 on this side. One is a little bit lighter. It's because of the low nitrogen it's able to absorb. Sometimes that's a good thing, sometimes that's a bad thing. In the case of 214 is a good thing because of this. Nitrogen and potassium, and related to that, are associated with bitter pit of Honeycrisp apples and some other apple varieties. And so we like to, here you have, this is one of our doctoral students that, and he's now he is now back in Oman, and sends me pictures sometimes of the many things that he's done. But he is using an X-ray instrument to measure the content of potassium and calcium in these different apples. This is Sarah Bauer, and she is also measuring, see this apple right here, she's measuring the content by shooting X-rays and reading out what the atoms, how many atoms did these X-ray hit. And so we can measure these potassium to calcium ratios right here. And they correlate very, very well with the bitter pit. So, and this is rootstock related bitter pit. These individual dots are actually means of 3 or 4 apple trees that had the same rootstock and with the same scion variety. So what does that mean in the hands of apple grower? Well, having bitter pit free yield in tons per hectare for certain rootstocks means, about, if you have the highest producing ones that are bitter pit free over the life of 10 years or whatever, it's about $247,000 per hectare. We measured this and did many projections on this. So if you have the best possible rootstocks that gives you bitter pit free apples like G969 or G214 or a few other ones that we have, you're able to have quite a lot more. So changing things. The climate is changing, and there is something that's called winter chilling requirements in apples. So if you don't have enough chill, if the buds don't feel enough chill, they won't break at the right time, and so you have kind of like a loss in production because the buds are not breaking. And it turns out that some of our rootstock, this is a brand new technology that we have in our breeding program. It turns out that some of our rootstocks actually increase productivity by lowering the chill requirement of the buds in the grafted scion. And you can see in this picture right here, this is, let me see, this is M9, which you can see a lot of the buds are not broken throughout the tree. And then this is G213, which is now widely planted in Brazil because of this trait. And does this trait translate to other rootstocks in our breeding program? Of course it does, because we have the genetics. And so this is looking at one of the experiments that we did, I think this was 4 or 5 years ago, where we were looking at bud openings at certain levels of chilling hours. There are other challenges in fruit production. You know, we have a lot of consumers that they don't want a lot of pesticides applied. So what can rootstocks do with pesticides? Well, there's a little critter that's called woolly apple aphid. And it survives in the ground during the winter time, and then it comes up, and in organic plantings, this is not a good thing to have. But what happens if you have a resistant rootstocks, it negates this living environment. And so if the winter kills the top, the aphids that are on top, they don't survive in the ground. And this has been a very valuable trait. And this is one of the traits that we've been studying in the past few years. So that's a correlation table showing that year over year of data, the evaluations that we're doing. See right here, some of these white things are the woolies that are growing onto the roots. And so it's another one of those valuable traits that are not quite new, but this genetics is a bit newer. I talked about fire blight. It's terrible. I've seen acres and acres of orchards killed by fire blight. This is what it looks like when a tree gets killed by fire blight. It takes a couple of weeks to do it. We studied the genetics over and over, several years, and with several different strains of the fire blight bacterium. But when you see it in the field, see these yellow lines right here? These are all fire blight affected trees that are going to die. This orchard was planted, I think it was Fuji planted on M9, and it had to be removed. And then this is actually an experiment that we planted in Utah, in an area that gets a lot of fire blight. Initially, the trees, you see a lot of trees, and they're happy and stuff, but then a couple of years later, we had our rootstocks interspersed randomly throughout the orchard. And guess what survived? The Geneva rootstocks. And that was just 2 years of growing. So having the resistance there means a lot to many apple growers. Something else that was new in the Geneva rootstocks was also the knowledge that they could survive replant disease in a much better way than traditional rootstocks. And so we have several studies that studied the interaction of this, what's called the replant disease complex. And we measured the yield differences. And you can do that by essentially killing everything in the field by fumigating, and then planting, and then an experiment, and then next to that field you plant another experiment, and you compare the two. And but what's interesting is you have 34% increase in yield when you have the most resistant rootstocks. And this is in Washington State. So this type of experiment has been repeated throughout the world now. This is in Brazil. We have the same thing happening in South Africa, in Europe. And so we've been trying to understand what is behind that tolerance of resistance of our unique germplasm. And we attributed to a combination of disease resistance and the ability of roots to grow a little faster, or to reproduce a little faster. That was work done by your student, Taryn, Brian Emmett, which was wonderful. We're just talking about it just the other day. But there's something that okay, so there was the initial selection that was done for resistance to Phytophthora and Pythium and so on and so forth, but there was something else that was also happening to those roots. We studied the genetic resistance to Pythium in apple rootstocks, and this is sort of like, it's very complex. It's very, very complex in terms of the resistance, and so we can perhaps use some of these loci to our benefit, but it's going to be difficult. But we're just continuing to work on this clever way, a very clever way that Rachel Lyso came up with, was to understand what the roots were doing, because some of these roots were actually changing the composition of the rhizobiota. And so what Rachel did is grow these rootstocks in sand culture, and then took all the percolates from that sand culture, concentrated them, and then looked at the metabolic compounds, or the metabolites that were found in there, and found that there was a relationship with what was growing. So she identified, they identified the benzoic acid as being something that was contributing. It turns out that benzoic acid, the QTL for benzoic acid matches with one of the QTLs in this population, in the content. That's the picture of Rachel, wonderful work. But looking at, so that was percolates. Looking at the roots themselves and the content, the metabolite content, how many of you knew that roots were producing caffeine and differentially producing caffeine? So here is, where is it? Where are you? Over here. Caffeic acid, right here. And you see some are more, these are individual rootstocks, by the way. And this is the correlation between all of, or among all of these different compounds. But caffeic acid, benzoic acid, and you see some rootstocks are producing a lot of this stuff, some very little, and so on and so forth. And so roots seem to be a lot more complex than I had originally thought of. We repeated this percolate experiment in a different way using aeroponics. Julie Carden just finished her master's, defended her master's, but and we looked at a lot more metabolites, and look at this. So significant differences in indole-butyric acid, IBA, or all sorts of other things among these 6 rootstocks that we tested. So we're in the process of understanding that. What's the relationship between that and the critters that are growing in the soil? It's a very, very complicated. And Julie did an awesome job at studying that. Moving on to a different topic is viruses. And this is what happens when you have one of our rootstocks, unfortunately, and the Sime variety has stem pitting and stem grooving. And you see, it's, most of the trees are like suffering because they're having a hard time. And then you have a few escapes here and there. And I think those are the buds that are at the very, very top of the bud stick that kind of have escaped the concentration of viruses. But this was a real problem that came up several years ago, and it affected one of our rootstocks in a major way, G935. And so we had maybe, we were producing about 3 to 4 million of these rootstocks every year, and people got scared because of this that happened. Well, one of the Sime varieties had every virus that you can imagine in it, and they put it on G935, and all of a sudden you see these trees that are not growing very well This is the virus-free, virus-load, and so we wanted to understand that. We planted a field trial and said, okay, is this something that is happening with all of our rootstocks or some of our rootstocks? And the understanding now is that it's with some of our rootstocks, and there are genetic factors that are associated with that. This is several different rootstocks that we were testing, and virus and non-virus free, and you can see that sometimes the line matches, which is they're insensitive to the viruses, and sometimes the difference of in growth is huge. So we like this. We don't like this very much. But so we leveraged, because shovelomics or digging around roots is very, very hard. We leveraged a different type of growing apple roots. We started playing with this back in 2016, 17, and then we have developed aeroponic systems. And this is a picture of our one of our greenhouses that had these aeroponic systems. We've done several experiments. If you want to learn about aeroponics, planning online, or else come up to Agritech and come and visit me. But that's how we did it. So we wanted to know what one of the experiments, and you'll see there are many more experiments. We wanted to know how many rootstocks, or what was the molecular mechanism of resistance. And this is the experiment right here, 10 reps of 10 things. This is what things look like. And it turns out that this is the same Scion variety, dirty and clean, that you saw earlier in the field. But now we've grafted it onto different rootstocks and grown the root systems in aeroponics. And this is G935. Look at this root system compared to the clean one. And then some of the other ones, some of the other material. So this is an ongoing process, but we have the first results from gene expression, and we're starting to learn what the differentially expressed genes are in the roots between virus resistance and not. So we are using those aeroponics. We've used it to study all sorts of other things, mineral nutrient uptake, pH, and so on and so forth. And I'm just going to quickly go ahead and show you. This is Dr. Ali Farkani, PhD work on pH, pH effects on apple rootstocks. Years ago, I said to myself, we need to go bigger with these and grow trees. And so we actually did that. We have a big aeroponic tank, and these are little dendrometers that I built in my office electronics lab, and they're able to detect the shrinking in the. Oh, this is what it looks like underneath. These are the root systems, and this is Davis Upchurch. We've had several people in this tank. Actually, we thought about using the tank. If this is going to get retired, we're going to build Mark II. We thought about using it as one of those, how do you call it, a stimulation sensor deprivation. So, but it's neat what we can see in the root systems. And we have lots of gene expression data from these, but these sensors that I built can pick up the cloudiness, if a cloud goes by, it'll pick up the increase in diameter of the tree in that, and we've used that to study over multiple months. We use it to study the physiology and the growth of the recent rootstocks. This right here that you see right there, that is actually a planned drought event. We have gene expression before, after, and during the drought event. These are other drought events that were not planned. But you can pick them up very quickly. And we're getting close to the last few slides, but look at this. We actually, so this is what happens during that drought event. We've really almost killed the trees. But they picked up again, and we also have apples that are growing on those trees. Nursery stock and viruses. Our lab actually, with a big collaboration nationwide, we developed these systems to a new system to clean the viruses up that use thermotherapy and cryotherapy combined together. And instead of taking, cleaning up, eliminating the viruses in 2 years, it actually takes about 3 months. And so we cut down a lot of that, and we're able to run this operation in-house thanks to Abby Nedro, who just had a baby. So congratulations, Abby. And with that, the micropropagation, we used micropropagation, and we did a lot of research on micropropagation because it's one of the ways where we've been able to make that change, a transformation from just a few rootstocks to a diversity of rootstocks. And it has allowed us to do this, to do containerized trees, to produce millions and millions of apple trees through micro. And so now the last few pictures that I show you are nurseries. These are this is probably a field of close to 2 million rootstocks and apple trees in Willow Drive. This is over in California at Sierra Gold. They are one of the biggest producers of micropropagated rootstocks. These are trees in Oregon at Kit Johnston. This is tissue culture derived trees at Agromillora in Oregon. And we have apple rootstocks all over the world. So this is a field day. Actually, it was a series of days, like 3 days, dedicated just to Geneva rootstocks in Brazil. Same thing in Mexico. And we've had the same thing in Europe and in other countries where these rootstocks have really transformed the landscape. I can tell you of productivity in those countries, so if you happen to come by in Geneva, I've taken my colleagues out for a sail. So between maybe May 25th and November 1st, come on over and I'll take you out sailing. That's one of the groups that was working with us, and apple rooster breeding is what we do. Thank you. I'll take any questions. Chris Watkins.
You mentioned your resistance to fire blight. Is it possible to grow commercially without using strip? Or are they still having to use strip?
They're still having to use strip. It depends on the sign variety. So some sign varieties are a little bit more resistant, but what the rootstock does, it keeps the tree alive during a heavy fire blight infection. So
just resistance to the
right, it keeps the tree alive. Sometimes even one little strike up top can travel all the way down to the rootstock, and if the rootstock is sensitive, and dies, you've lost the whole tree. So you don't need a lot of fire blight. In fact, some people walked into the field in fall, and they see all these purple leaves, and they go, "Oh, yeah, things are fine." We didn't see any fire blight. Then next year, trees don't come back up. So I've seen that. Yes. Yeah, your question.
Thank you for the talk. So you granted a lot of rootstock sign combinations. Have you ever seen incompatibility? Especially for the wall inducing rootstock sign combinations, because I heard partial incompatibility is one of the mechanisms that rootstock induce sign the wall.
Oh goodness. So we have done a lot of research on that. Yes, and we many and I don't like to talk about incompatibility. I like to talk about affinity. There are terms that kind of describe the same thing, but they're a bit different. I don't think there is the dwarfing character is related to incompatibility or affinity. Okay. Because we have many dwarfing rootstocks that have good affinity with certain science and then not very good affinity with other science, and they break. So dwarfing is not related to the affinity for the most part. I mean, there could be a little bit of that going on, but for the most part, no. It's a different physiological thing, and we have studied that process. Auxins is a big deal. Auxins are a big deal in the formation of the graft union. We published a few papers on it, and we can talk later about it. Yes, Yusuf.
Thank you, Gennaro. You mentioned about the effect of the rootstock on the shedding requirements.
Yes.
Yeah. What's the mechanism? How?
So yeah, we actually have a grant going on, and we're studying it. We're studying the mechanism. What's your best guess? Okay, that's also my best guess. So we've noticed that initially that hormone balance changing with different rootstocks is different, but then it's probably a little bit more complicated than just hormones, because hormones might be setting up the bud, but what's happening in the bud that is set up in the fall that has less killing requirements. Okay, so you may have some hormone signal or something like that that's happening, but then what's happening internally in the bud, and what's that mechanism? So you have to kind of think about that. Yeah, the effect is real, and it's going to be more and more important as climate change, as apples are planted in places where you don't get enough of that chilling requirement. Students, come on. Albert.
Thanks, Gennaro. General question for plant breeding program itself and your apple rootstock breeding program. A lot of plant breeders say, well, here's a new genomics, we can gene edit, and on the scion, there's the Arctic frost. Any thoughts on, as we get more genomic data, implementing some of those techniques into the apple rootstock breeding program?
Yeah, that's a great question. So gene editing and apple rootstock breeding. So as I showed you some of those slides of the QTLs or the loci that are involved in certain traits, gene editing, you can change things once or twice in the same process, in the same individual. So you have to have targets that are that important and that are that strong of an effect. We haven't found those targets yet in apple rootstocks, but it is possible that with dwarfing, if you know the target, probably gene edit and take a rootstock that has certain properties and just change it slightly. So it's possible, and I think that eventually that might happen, and it might be an easier way to change an already established rootstock to do that. But for example, fire blight in M9, it's pretty loci. And the combinatorial statistics of those events, because you have to transform it or transition, do a transformation in such a way that it's not transgenic. It's way high, and it's cost millions of dollars to do that. And then you still have to evaluate it for what, 5 to 10 years in order to see, is it really dwarfing? Is it really productive? Or did the changes that we make actually mess something else up? Or is there a pleiotropic effect of changing these 4 base pairs in this? Okay, so Albert is another very important person in the breeding program. Albert and Emily. I mentioned Emily, but Albert stepped in. So they represent CTL, who are the managers of the intellectual property of our breeding program. And as such, they deal with contracts and all sorts and exchanging plant material, make sure that all the T's are dotted. No, not, that's my Italian. Yeah, very good. That's my Italian. But yeah, so when I had that slide on the technology transfer and the receiving of that award, the technology transfer, that it was helpful to have people, very, very crucial to have people that did all the contracts and the testing agreements and so on and so forth in that. So before Albert, there was Jessica Liga, but we are very, very grateful for their help. Yep.
So when you're breeding for specific traits like fire blight resistance or like William said, is there a quantitative way to determine when you're breeding for like something like that, but then at the detriment of other traits like food size or like nutrient mineral uptake? Is there a way to quantify when that stops being?
I think the best example is mineral nutrient uptake. Yeah, and there is, you have to have a lot of data on that. But yes, there is a way where we've seen some of these correlation coefficients among nutrients sometimes go negative. For example, potassium. We increase potassium in the fruit through the rootstock, and you see calcium drop off many times. Not always. We like both to be up, but there are those instances like that, or magnesium and things. So there are sometimes instances where you select for something and then something else is at a detriment, and you have to play with that, or you have to create enough genetic recombination events and enough combinatorial events to overcome those restrictions. We also, I didn't show you this, but we have a huge program in genomics and marker assisted breeding and all of that. It's boring stuff. It's fun, but I didn't want to have all of that in this presentation. That's another 10 hours. So, yep.
It's an incredibly impactful and impressive program that you have here, and all I want to do is say congratulations to you and the team. It's really amazing what you guys.
Oh, I deserve very little credit. The team and especially the inceptors of the program with Herb Alwinkle, Jim Cummins, and he's still alive, by the way, and he was pruning apple trees last year. Terrence Robinson have done, I stand on the shoulders of very, very good, just visionary people. So imagine 60 years, almost 60 years ago, thinking about this. And they did. So my thing is, I'm trying to think for the next 60 years. And so we're doing, climate change research and some of these other things. But yeah, it's breeding for something that takes so long to be implemented and planted. It's got to be crazy to do that. Okay, All good? Well, thank you for listening to me. I appreciate it.
This has been a production of Cornell University, on the web at cornell.edu.